We present an approach for analyzing the parameter space of spatio-temporal simulations and finding a parameter space partitioning. We propose to use linked views covering the similarity space of the dataset as well as its parameter space. We want to find a clustering in the similarity space and investigate the induced partitioning in parameter space. The temporal evolution plot gives an overview of the whole ensemble and its evolution over time. Each line represents one ensemble member. The similarity embedding shows the similarity between entire simulation runs, where each run is shown as a point. In both visualizations, the dissimilarities between runs are encoded as distances. We visualize the hierarchical clustering of the similarity space in a clustering dendrogram, where a horizontal line symbolizes the pruning level. To create an overview of the parameter space, we use a parameter sample embedding using MDS. The large dots show the barycenter of the clusters induced from the similarity space. Our main visualization for the analysis of the parameter space is based on hyperslices, which we adapt for the analysis of parameter space partitioning. It shows axis-aligned slices around a focus point that can be changed interactively. Hyperslices allow an undistorted view of the parameter space, the direct deduction of parameter values, and most domain experts are used to the slicing of volumes. The values of the focus point are shown on the diagonal below the parameter names. A single slice viewer can be enlarged for a more detailed analysis. The gray lines visualize the values of the focus points for the parameters shown on the axis. We show a projection of the simulation runs by using empty circles. If the point lies in the slice, it is filled and encircled in white. Tooltips show the names of the runs in the corresponding cluster colors. The distance to the points with simulation results is represented by varying the saturation of the cluster colors. It is possible to select the parameters that should be included in the hyperslicer to keep the dimension as small as possible. The selection is guided by a calculation of the correlation between the parameter values and the simulation outcome. To facilitate the navigation in the hyperslicer, a cluster can be selected in the parameter sample embedding for further investigation. The focus point in the hyperslicer switches automatically to the segment induced by this cluster. We can project the boundaries of the segment and visualize it with dots in the same color as the cluster. The use of Boolean expressions allows for more detailed analysis. Single ensemble members can be visualized using spatial visualization methods like volume rendering or axis-aligned slice viewer. Now we want to compare our hyperslicer visualization with a scatterplot matrix and a parallel coordinates plot. Here we see the results for a randomly sampled synthetic dataset. We can see that the blue and the red segment are separated by the choice of parameter C. However, identifying the extent of the purple and the green segment is impossible in the parallel coordinates plot, because the segment boundaries are not parallel to one of the axes. Due to the high number of points, it is also hard to obtain an understanding of the geometric shape based on the scatterplot matrix. In the hyperslicer, we can clearly see the diagonal boundaries for the selected focus point. By enabling the projection of the green cluster, we can see that it is also present for other parameter values. We now change the focus point interactively and see how the segment's shape varies. This interactive exploration, together with the projection, enables us to verify that the segment is bounded by a diagonal in the parameters A, B, and C. Next, we show the parameter space analysis of a spatio-temporal simulation of a blood flow dataset. The temporal evolution plot can be used to select a relevant time interval for further analysis. It is also possible to select the linkage algorithm used for the clustering. We select a suitable pruning level in the dendrogram. In the parameter sample embedding, we can see that the groups in parameter space do not agree perfectly with those found in similarity space. We investigate the parameter space in more detail in the hyperslicer. To limit the parameter space dimensions for this analysis, we observe the correlation between the simulation outcome and single parameters. We find two parameters with a very small correlation that we exclude for the following analysis. In the next step, we can further investigate the clusters one after another. We start by selecting the red cluster, which forms a group in the parameter sample embedding. We want to confirm that the region is connected in parameter space and identify its parameter values. We could project the segment by selecting complete or inserting complete as a Boolean expression. We can show the complement by inserting a not in the Boolean expression. The texture now shows the region where the segment cannot be found. We can see that there are whole regions of parameter ranges where the cluster does not occur. With this technique, we can clearly identify the parameter region of this segment. It only occurs if Poisson boundary conditions are turned off and the length is large. We turn on the uncertainty visualization to see how reliable the identifying boundary between the different segments is. In this case, the uncertainty is small. Last, we select a single ensemble member for in-detail analysis with a spatial visualization method. For this dataset, we use a direct volume rendering. We compare the simulation result to a simulation result of another cluster to understand the differences. Then, we observe the volume at different time steps.